and welcome to this online presentation by FluoGuide of results from the FG001 clinical trial. My name is Thomas Petersen. I'm helping facilitate this presentation today. Here today with us we have the two gentlemen, as you may know, from FluoGuide. That is Morten Albrechtsen, the CEO, and Andreas Kjær, the Chief Medical Officer. And furthermore, we have a special guest, I think I could say, that is Jane Skjødt Rasmussen, who is a consulting neurosurgeon and principal investigator of the trial. I'll not take much more time. I'm here together with Morten, and I'll hand over the scene to you, Morten, and you have prepared a presentation, but maybe you'll introduce the other presenters a bit more.
Yeah, thank you very much. Thank you, and welcome for the presentation today, where we are extremely proud and very glad that you, Jane, will join us. Jane is the principal investigator for our trial FG001 in aggressive brain cancer. Jane is also a consultant neurosurgeon at the Rigshospitalet in Copenhagen. So this is the first data that will be presented, and that were presented here in the weekend in Bergen in Norway, and the Scandinavian Neurosurgeon Society conference. First, before Jane will go into the data that were presented in the weekend here, there will be a few background slides to get us all up to speed on brain cancer and how it's treated. And follow the presentation, there will be a discussion, and you're very welcome. We have already received some questions, but please be free to submit questions to Thomas, who will organize it, so we are sure that we address all questions that is being asked. After the presentation, I'll just round up with a short update on the clinical trials, our plans, and what is ongoing. Really, with this word, we're quite pleased to give the word to Jane Skjødt Rasmussen, that did the award-winning presentation abstract in Norway. So, Jane, the floor is yours.
Thank you very much. Yes, so this is the results are quite new, but nonetheless important. So first, I'll just give you a small insight on what brain tumor is, because it's actually there's a large variety of what kind of cancer we're talking about. So the primary cancer that we have in the brain is derived from the supporting cells, also called the glia cells in the brain, and one of the most frequent one is the astrocytoma. And the astrocytoma can be divided into grade 1 to 4, where grade 1 is a very benign one, and grade 2 to 4 are more and more aggressive the higher number you have. And then you have other types of tumors in the brain that I will not get more into. So if you could get the next slide, please. So here's an overview of the primary brain tumors. And you can say, as you can see, there is a large number of meningiomas, which are benign tumors in the brain, but there's also a great representation of the glioblastoma. And the glioblastoma is the most malignant glioma of all the gliomas you can have in the brain. So in general, if you talk about primary brain cancer, most people will be thinking about glioblastoma, and that's also the one that we are investigating now. You can also call it astrocytoma grade 4, and it can have some other numbers on it, which is it's not something that you can say in advance, but that's something that has to do with the subsequent treatment. And the next slide, please. So if you have a benign tumor, you remove it completely with surgery, and that's often possible because they are more demarcated towards the brain. If you have the slow-growing tumors that are also infiltrating, meaning that there's no sharp border between the brain and the tumor, then they're also surgically removed, but then they are not giving other treatment up front, but they are followed, and sometimes they will progress to more malignant behavior, and at that time they will have renewed surgery and also oncological treatment. But for the faster growing tumor, which are the one we are talking about here, we will always do surgery and always supplement that with other therapy such as radiation, chemotherapy, often in the combination that is known as the STUP regime that was developed back in 2006 and is still of use. And then you can also combine it with some new experimental treatment, but that's not something I'll get more into here. So basic principles in treating the primary brain cancer is surgery and then subsequent oncological treatment. Can I get the next slide, please? So when you talk about surgery, you can talk about excision, which is complete removal of the tumor. You can talk about resection, where you make the tumor much smaller and then you will give treatment afterwards. Or you can sometimes just take a biopsy, and that can be in cases where patients are too fragile to go through a complete surgical removal, and then they will have a biopsy for the specific diagnosis and then palliative treatment. So after the surgery is done, you will send the tumor for grading and histopathological diagnosis. That will take around 5 days, sometimes a bit more. And then the after treatment with the neuro-oncology will start within 4 weeks, typically. And next slide, please. So that was a bit of the background. So the tumors we're talking about here is malignant glioma, and most often that will be the grade 4 tumors that we're talking about. So this is the first in human study of this novel UPAR targeted imaging agent FGD01, and it's used for visualization of malignant glioma during surgery. Next slide. And these were the closures that I also showed in Bergen, that I'm supported. The study is supported by FluoGuide, but otherwise it's I'm not on the payroll such as. And next slide, please. So the aim of the study that we have been conducting was to investigate whether this UPAR targeted optical probe can visualize the malignant glioma. And what does it mean when we say UPAR targeted? Well, the UPAR is a protein that binds specifically to the cancer cell, and this has been had an add-on of this fluorescent dye, and then you can see it in the microscope that's used for most well, it's a filter. It's in near infrared range, and that's a filter that can be seen by most surgical microscopes, since it's also used for visualization of vessels during surgery for vessels anatomy in the brain. And this design was an open-label, non-randomized dose-escalating study, meaning that we had to see if it was a safe drug and if it was a drug that actually worked. And next slide, please. And here is just an overview of what patients we had. So distribution with more male than female, that's the normal distribution. The mean age, 58 years. And height and weight and body mass is also included here, which is, so you can see that there's the normal range of that as well. And next slide. So how did we do this study? Well, we took patients. We started out with 1 milligram, and we ended up injecting the drug into a total of 40 patients. And then we did surgery, and we were determining how good the drug was to discriminate tumor from brain and measure that as the tumor to background ratio. And what we could see during the study was that from 16 milligram and further on, there was a consistent visualization of the tumor. And next slide, please. We were very meticulous in recording any events during the study, and we found no serious adverse events, which is a very good thing. And with the adverse events, there were only a few minors, grade 1 and 2. And next slide, please. So tumor to background ratio, that's the way we like to see how good is the drug to discriminate the tumor from the brain. And the reason why this is so important is that when you do the surgery and you have the normal microscope in white light, it's difficult to see the border between the tumor and also to see the infiltration zone. And it's actually the infiltration zone that is important for the surgical for the surgery, because that's where the tumor will regrow from. So the infiltration zone is the important, and that's what the FGD1 helps us be sure at where it is. Yes, and next slide, please. So this is a video of the surgery, and the fluorescent, very bright, shining in the depth, that's the tumor. And then you can see we are 2 surgeons working actually, and it's the tumor is kind of pushed to the center, and then there's the suction that kind of works in the infiltration zone, so to release the tumor from the brain, so that we eventually could take it out. And next slide, please. And just to ensure that whatever was fluorescent was in fact tumor, we took some samples guided by the light and sent that for pathology. And even though it was a low number of samples taken, we found a very good specificity, which is very important, and also very high positive predictive value, which is also very good. So these numbers are good. When we find something that fluoresces, it's tumor, and that makes the drug reliable. And next slide, please. So what we could conclude from the study was that it's safe, it's well tolerated. We found out by our imaging and calculations that 60 milligram was the one that provided the excellent image, and that was high contrast during surgery. And we confirmed our findings with histopathology. So now we're ready to go on with phase 2 trial also for malignant glioma. And what is also of great interest to explore what value can this bring to surgery in other types of brain tumors. And as you can see, we are only talking about 1/6 of the tumor with this, but there are others that could be extremely interesting as well to investigate. So next slide, please. There's a lot of people involved in this, so I just wrote a little thank you slide for those people, but actually we also need to thank the patients who are willing to enter a study like this. So that was my presentation.
Thank you, Jane, very much for the presentation. I think we open up for questions now and discussions.
Yes, thank you very much. As some of you will see, there is a small box in the lower right-hand side of your screen. If you're on a mobile screen, you may have to scroll a bit up and down before it appears, and it may also be a bit delayed. We welcome any questions to be added to the conversation, and today I'll start off with asking a few questions. Let me first see if it appears that anyone is asking any questions. It's not yet, but feel free to do so. Jane, I have one question. Like in layman's terms, how would you say that FG001 performed? We have, a lot of people here, and very good, thorough explanation. But in everyday language, how would you say it performed?
No, it performed really well. I mean, what you need as a surgeon is that you need to be sure that you take something out that is actually tumor, and the more certain you are of that, the more you can actually help the patient by taking away the cancer, because you also need to be very careful not to do any damage to the patient. And the important thing about these tumors, these gliomas, is that they are infiltrating, so there's no sharp border. So we need some adjunct to help us see where is the border, where is the tumor, where is it normal brain, because that's not visible in a normal microscope in white light. So we need some fluorescent drug or other drugs that can help us visualize for certainty where is the infiltration zone. because it helps with the survival, that you can remove, if you can remove the tumor more efficiently and then give them the treatment afterwards, but that prolongs their lives if we can give them a better surgery.
Thank you. I can see the questions also relate to other areas that this particular study, so I'll just help the audience to remember the agenda of today. The agenda of today is that first now we have the conversation about the study, and then later I'm sure we can ask Morten and Andreas some more broad questions. So I'll try to focus on the questions related to the study first. Another one for you, Jane. Would you use it, that is FG001, even if it was not something you were involved in doing research on?
Yes, I would be happy to use it, actually. I think nowadays the administration is very, the administration of the drug up till now has been very much handled by research people. But if it was ready for use, so to say, for the general nurse, I would be happy to use it. Yes.
All right, thank you. And I don't know exactly who is the right one to answer this question, but what does it precisely mean when you say that it's safe and well tolerated? Jane, Morten, I don't know.
Andreas, I think.
Andreas?
I can take that. Yeah, a part of Jane explained the study so well, and apart from that, it lighted up, and that you had good contrast in the tumor, and that what lighted up was actually tumor. The other part of the first in human study will always be safety and whether it's well tolerated. So these patients have all been monitored very closely. And that means ECG, blood pressure, blood samples, all these vital parameters, and to see if they had any reactions on the drugs. And that was, as Jane mentioned, no serious adverse events and only very few very mild adverse events. One thing that is important to understand is that these patients that Jane is operating on are quite sick, and when you do brain surgery, there will also be some side effects from the surgery. So it's important to see that as adverse events and then to follow up whether could this be related to the drug or not. And as mentioned, only very few, actually only 4 that was very mild. And examples of that could be one was pain from the catheter injection site, has nothing maybe really with the drug to do. And then there was one patient having nausea. And maybe that should be mentioned. We could not rule out it was related to the drug, but these patients actually received a drug that is commonly used now as a standard 5-ALA. So, and these were given in close relation to each other. So whether it's really one thing or another, we cannot say with absolute firmness, but what will be done in the phase 2 study is actually that patients will either receive FG001 or the comparator which will be 5-ALA, and then there will be built in a score on nausea. So we will that is one way we will try to go into this. But as you can see, it was 40 patients. There was one very mild, and maybe it's not related to the drug. We cannot just rule it out. So very safe. And is this important? Yes, we could also see that there was no relation. Actually, the very few mild adverse events were at a very low dose, the majority of them. So normally, if it was the drug, you would expect that the higher dose, the more you would see. So this is a very good indicator that it's very safe, and it didn't restrict us in the dose. Jane mentioned that it was 36 milligrams that will be used, and that was because you could increase the dose. There came no more side effects, and at the same time 36 milligram was where the highest contrast was seen. Going further to 48 didn't increase the contrast more. So this is kind of now fixed, that this is the dose we will use going forward. And that was what is called the dose escalation part of the study. So a lot of valuable information, apart from the most important, that Jane thinks it looks really good and it can guide her, and that it indicates what is cancer, but also these what dose should one move forward to, when should it be given, and if is it safe and well tolerated.
So yes, I mean, this is the overview of the clinical trials we're running. We have now completed the 2A trial in aggressive brain cancer, and the part 2B is being in preparation. We have the less aggressive brain cancer study that will be started out after we have submitted, done the paperwork for this 2B, and as well the briefing book for the FDA. And then we have a lung cancer is ongoing. We have all the green light for it. This is ongoing. So this is very much the two set of studies we are going after. So we have brain and we have lung as two indications we are focusing in on. And in terms of the overview of the clinical trials, a couple of things. I mean, it's quite important for us that we learn as much as we can for the trials we're doing, preparing us for the next phase and address the risk and be as predictable as possible. So we have all the time since we very start, we have been long-term oriented, so we want to get as predictable to the market with the highest, most benefit product we can. So the couple of learnings have been in this phase. We have the result, of course, which is very nice that we see everyone that we can use it to guide surgery. We have seen that it worked very well across equipment and given setting. We have seen that the file from OnTarget they had on their for ovarian cancer, their HL 38 was published before Christmas. There was a lot of learning from that file, and then we have had discussion with other key opinion leaders than Jane in terms of the design of the trial for the next phase. And then last but not least, we have all the time looked at let's say adding more benefit in the documentation of the endpoint. The more benefit we add to the endpoint, the more clinical value we can provide or proof we can provide, and the higher price we can get. So it's very valuable for the company and the product that we can document a benefit. On the other hand, if we go for too ambitious benefit, then we actually it would take us a lot of time, a lot of money, and it would be more risky. So it's very much a balance between the optimal benefit and the optimal time and cost. So what we have agreed to is that we step a little up in the benefit, and that would be the proportion of patient that will have a clinical benefit for the change in surgical strategy due to our drug. And that actually a clear benefit for the patient that we prove this rather than saying just light. So it's a step up, but at the same time, the study will still be this 48 hours inclusion into the trial, and that is one decision we have made for the phase 2B study commencing now. The other thing has been that we have seen that in the trial here, as Jane mentioned, the 5-ALA is giving some nausea for it's known for the product. We have not seen that for the evening dose of FD1, and that actually makes us to believe that this is related to the 5-ALA and not FD1. And therefore, we have in the next study we will do it in 2 arms, so we separate the 2 drugs. In this study we did here now, we are giving on the same patient. They have had the 5-ALA, actually all of them. They were said that it should be the standard treatment, but in this case it was 5-ALA. It will be Sweden and Denmark still, and we still have an expectation of completing it in this year 22. With this data, we can this will be a mini phase 3 study, and we can go straight into a phase 3 study. And still, we also remain our timeline for that one. It will be half US, half European in terms of site. And then we will start, as mentioned, the less aggressive brain cancer. That's important because the 5-ALA does not work in cancers except for this high-grade glioma. So that's one differentiating benefit that potentially could be for FD1. We know already now that it would work in the meningioma, where we had one patient in this trial that was in, and one patient, of course, is not approved, but it still is reoccurring. And then we have the lung study ongoing right now, and will also be completed with result this year. So this is very much your overview, but I think that the slight change is that we add the benefit. We add some patient from 12 to 24 patients, and we separated in 2 arms to be able to prove the safety of FG1 that we have seen this trial more clearly. And I think that's yes, it's just the sales potential for the 2 indication. As you can see, I mean, lung is more patient with lung, but still the brain cancer is quite interesting. The lung is very much visualized in the red eye report, where the brain is more visualized in the APG report on the coverage we have. They're both very important, and a lot of patients out there need better guidance. So with that, I think that's very much what I have to say. And